Hello everyone and welcome to our video. I'm Caroline and I'm going to be talking about the book The Sports Gene by David Epstein. Before we jump into the video, as you may be able to tell, I'm a double helix DNA molecule, and you will meet my other DNA friends later in this video to tell you about current research. For a little background, as you may know, DNA is made up of sequences of nitrogenous bases that code for genes. However, in 3 million base pairs, a single mistype could cause a problem that is minor or life-threatening. This book focuses on how genes play a role in an individual's particular athletic abilities, their motivation, and their endurance, which overall contributes to an athlete's success in a particular sport. We found an interest in Chapter 15 titled The Heartbreak Gene, which we will explore in this video presentation. Epstein opens the chapter with a story about a runner named Kevin, who had a mutation called hypertrophic cardiomyopathy, or HMC, which hinders the flow of blood to the heart because of the thickening of the left ventricle walls, which you can see here. This is one of those examples of an extreme outcome of a mistype because Kevin suddenly dropped dead without any warning signs or symptoms after a big race. Next, Epstein had talked about other genes like APOE gene, COL1A1 gene, and SCN9A gene. Mutations in these genes can also have fatal outcomes. For example, the SCN9A gene can have a rare mutation that causes someone to feel no pain, thus leading to infection from not responding to injuries, and ultimately to an early death. But one can also have a certain variant of a gene that puts them at a higher risk for certain diseases than others without the variant. For example, the APOE4 variant can leave a person at a higher risk of getting Alzheimer's disease than people without it. The COMT gene was also an important gene mentioned. My DNA friend, Olivia, will be discussing the details and characteristics of this gene variant and how it can be utilized for gene therapy and its treatment towards mental health.
Woo! Just learned that new magic trick. Hi everyone, my name is Olivia, and I'm here to provide further information on this very useful variant. The COMT gene is very famous for its role in pain modulation. It is involved in metabolizing neurotransmitters in the brain. There are two common versions of this gene, known as VAL or MET. These names come from the specific part of the gene sequence that codes for the two amino acids, valine and methionine. These two versions are also known as the warrior or the worrier, due to the effects of having one of the versions. The MET version of the COMT gene is not as effective at clearing out dopamine, and it ends up leaving more of it in the frontal cortex. So from this diagram to the right, you can see that when you have VAL, it increases the activity of the COMT enzyme, which therefore decreases the amount of dopamine you have left in your brain. And it would be the opposite for the MET. So it would decrease the activity of the COMT gene, but leave you with increased levels of dopamine. If a person has two MET versions of the COMT gene, this can result in the person having a better aptitude for cognitive brain tests and memory, but have an inclination to worry more and increase in anxiety, making them worriers. People who have two copies of VAL versions do not perform as well on cognitive and memory tests, but act as the worrier, having a higher pain tolerance. On the pie chart in the right, this is the frequency of the two variants. GG and AG both represent homozygous VAL variant, while AA represents the MET variant. More people in the population are going to have VAL variant over MET variant. Multiple antidepressive treatment methods are used in the clinical field. Research in this field is continuing to expand to avoid the addictive properties of antidepressant pharmaceuticals. This research has explored the association of the COMT RS4680 polymorphism with the treatment response in major depressive disorder patients. Major depressive disorder, or MDD, is a worldwide chronic mood illness characterized by cognitive dysfunction, which includes side effects of low morale, weight loss, and lack of pleasure. This currently is being treated through antidepressant drugs and electroconvulsive therapy, or ECT. Genetic factors are suggested to contribute to the pathogenesis of MDD, as well as involvement in the therapeutic effects. Moving on to a lighter note, my DNA friend Gabby is going to talk a little bit more about the COMT gene and how it affects the neurotransmitters in the brain.
Whoa! Hey there guys, my name is Gabby, and I'm going to be talking to you about the further effect of the COMT gene on neurotransmitters in the brain. On the COMT exon, the substitution from G to A changes the amino acid from Val to Met and the membrane-bound COMT protein. This missense mutation, also called the RS4680, leads to low activity of the COMT enzyme. Depending on the genetic variant of the COMT gene, the enzyme that is produced metabolizes the neurotransmitters dopamine, epinephrine, norepinephrine at different rates. The enzyme, which is coded by the COMT gene, ultimately plays a crucial role in maintaining a normal neuroendocrine environment, ensuring the stability of the levels of these neurotransmitters. This metabolic process occurs when the COMT enzyme adds a methyl group to the neurotransmitter, ultimately making the neurotransmitter a different molecule. This new molecule can then be disposed of in the cell because it is not of use anymore. The G allele, or Val, codes for higher COMT activity, meaning it's going to break down the neurotransmitters at a faster rate. This, in turn, is related to lower dopamine levels. On the opposition, the A allele, or the MET, has lower COMT enzyme activity. This lower COMT enzyme activity allows there to be a more dopamine present in the brain at once, allowing for a happier state of mind due to the presence of endorphin. The research proposed that the COMT RS4680 might be considered a candidate gene for predicting the pharmacological response to antidepressants of selective serotonin reuptake inhibitors. In conclusion, this meta-analysis indicated that the RS4680 polymorphism is associated with an antidepressive treatment response in a specific genetic model. More research must be performed to verify this conclusion. And that's the end of our presentation regarding the COMT gene and its use of gene therapies for helping mental disorders. Now, here on this slide, you'll see our images cited. And on the next slide, you'll see our work cited for this project. Thank you so much for listening to our project. Next up, we have behind the scenes.